inviting us. I think this is really nice to know you all with your children. And I would also like to take this opportunity to thank you very much, because we received the first research support from you already. And we'll use that to prepare iPS cells from 2 patients that we have already there, and to support the natural history trial. Yeah? So this is something that we come on. But I would like to start a little bit. We have 3 parts of the talk. I will do the introduction and show you a little bit what we have done so far. And then Lario will present what they do, and then we have some first data with the compound in mouse models. And for the Lario presentation and the mouse models, we would like to stop the recording because these are all unpublished data, and you never know what happens with these data if they are in the internet somewhere. And then we can restart the recording after we come to the natural history trial, or when we come to the natural history trial. Okay, so this is one of the calcium channels that is affected, and there are lots of calcium channel subunits. The main subunit is the alpha subunit, and CACNA1E also encodes one of the alpha subunits, and it encodes the calcium channel CAV2.3. And this is the so-called R-type calcium channel. And there are 3 types of presynaptic calcium channels. They are shown here. Can you see the mouse or no? So I take the laser pointer and it should work. Okay. Now you can see it, right? Yeah. Okay. So these are the 3 presynaptic calcium channel types. The PQ type is the main type. The N type is also a major type, and the R type is a smaller one. Or has a smaller contribution, and I will show you in the next slide the function about it. And it's called the remaining current because there were specific blockers from spiders available, and when you blocked those 2 channels, this was the current that was remaining, that was still there. And there are additional subunits like alpha-2 delta and beta, which are also part of diseases sometimes, and the gamma subunit is probably not really associated with calcium channels. Okay. So what's wrong? Okay. These are the diseases that are already linked to calcium channel subunits. This is the disease your children have. And there are parallels, for example, with Timothy syndrome, which is a combined, very complex syndrome, which we shouldn't discuss in detail, but the common thing is that they have very similar variants in that type of calcium channel that also cause a gain of function, and we'll come to this in the next slides. Okay, 2 nerve endings which come together at the so-called synapses, and this is the pre-synapse, so this is a neuron that projects to the other neuron, and this is the post-synapse, so the neuron that is receiving the transmitter. And calcium channels play an important role, not only CAV2.3 but also others in the presynaptic calcium entry. Yeah, so you can imagine that signals are coming along this nerve here, and they lead to a depolarization of the membrane. Yeah, and then the calcium channels open and release calcium into the presynaptic nerve terminal. And this calcium is very important to do this kind of process here, and this is how transmitters, so the red dots, they are the neurotransmitters, and the neurotransmitters are packed in vesicles, and these vesicles are then melting with the membrane and releasing the transmitter into the synaptic cleft. And then this transmitter can bind to these postsynaptic receptors. And therefore, calcium entry is crucial. And you can now see what the function of this channel is. So this is presynaptic calcium entry. And now this is work done by a collaborator of us who has done this work in 2003 already. and he has compared mice which lack the CACNA1E gene, so which lack these calcium channels, with normal wild-type mice. And under these conditions, they already blocked all the other calcium channels that are presynaptic. And what they could see then, that only after a tetanic stimulation, so this little symbol here stands for very high frequent stimuli. Yeah? So every time you see a vertical line, this vertical line is a stimulus. And this is called a tetanic stimulus if you do several ones in a row. And this is also happening in epilepsy. Yeah? That we have this tetanic stimulation endogenously, and here it's exogenously. And you can see that there is a remaining postsynaptic current. This is now a current that is measured in this cell here, and that is flowing in through those receptors here. And you can see that this is increasing and then decreasing, and the first phase is this here. This is just this little one. This is slowly increasing, and with each stimulus you get a smaller increase here, yeah? And then you get this tetanic. And in CAP2.3 knockout mice, this is missing. So they play a very important role for this release of calcium at high frequency stimulation. Okay, so the starting point of finding this disease was by our collaborators, Heather Mefford, who led the group, and Katie Helbig, who did all the recruitment of the patients. And there were altogether 30 patients recruited between an age of 10 months and 25 years. These were 14 de novo missense mutations. De novo means, as you probably know, that you as parents do not carry these variants, but only the children. And the phenotype were severe intellectual disability, developmental regression, mostly pharmacoresistant epilepsy, so only a few ones responded to pyramid. Then spasms, focal tonic seizures, and focal impaired awareness seizures, and generalized tonic-clonic seizures were the seizure types. There is an additional hyperkinetic movement disorder. The children move, many of them all the time, and there is for many a postpartal hypotonia, so less tonus, and the muscles are weak and flexing. Congenital contractures is also one type, and spastic tetraplegia means that the limbs cannot be moved, and that there is a spastic increase of tonus. Also, early death is one of the phenotypes. And then were 3 patients with truncating variants. I think this is less clear, and this probably none of your children has a truncating variant, or are you aware that there are some with truncating variants that have a loss of function? Probably not. Yeah. So and for me, it's less clear if it's really the triggering factor. Then there was a second publication coming out later, and that was a broadening of the phenotype because they had no seizures. And there were also a few ones in the gene measure database, and you can see that specific variants cause seizures. Yeah? So the epilepsy are the blue dots here, and those are the variants that are clustered in the so-called S6 segments. Yeah? So every calcium channel has 4 of these domains, and these domains each have 6 transmembrane segments, and the segment S5 and S6, those 2 here, form the ion channel pores, so form the conduction pathway where calcium is going through. And the S6 segment is particularly important. It's the gate of the channel. Yeah? So if you have that channel like this through the membrane, yeah, and calcium is flowing this way, the gate is opening and closing. And right within this gate, there are the variants. And the interesting thing is that here it is again, yeah, so these are the variants, and the calcium entry is facilitated. And this is what you can see here. So this is the so-called activation curve. Plotted is the activity of the channel, which is called the conductance here on the y-axis. And on the x-axis, you have the voltage that you have to apply that the channel opens. And for the mutant channels, you need less voltage to open them. And that's why this is called a gain of function. So they open more easily, yeah, upon membrane depolarization. And then they stay open longer, and you see this best in this channel here. You see that the decrease of the channel is slower than here for the wild type. WT is wild type, and these are 3 variants. They are all located here in this 6th segment of the second domain. And this slowing of the inactivation also causes a gain of function. So this is an increased calcium flow through the channel. Okay? Good. And the next is just repeating this. This is this channel here. This was a contribution from Gerald Zamponi, who studied this variant, which is located in the S4-S5 loop. And this S4-S5 loop is also communicating with the gate probably. And that's the reason why there is the same facilitation of activation. Okay, then there is a second disease which is linked to CAF2.3, and this is really interesting because as Henning and Tom will later point out, this is also then an interesting target for these antagonists of CAF2.3. This epilepsy group is much larger because the disease is longer known, yeah, so I'm not sure if it's really more frequent, but that's something that time has to show. But at the moment there are many more people known with the CDKL5 disorder. It's also a type of developmental and epileptic encephalopathy. What the case is, these are now the 4 domains, yeah? So each of these domains has, again, these 6 transmembrane segments, and between the first and second domain and in the N-terminus, there is a phosphorylation site. And this phosphorylation site regulates the activity of the channel. And when you remove the phosphorylation site, and this is when you replace the serine by an alanine, then CDKL5 can no longer bind. And then you get this slowing of the inactivation again here. Yeah? So very similar as to what we saw before for the variants. And you also see an increase in current here. So this is the current size averaged about over many cells. And then you can see that the average is increased. So there's also an increased calcium current, and this can be also treated by CAV 2-point antagonism.
So we talked a lot about basic science now, so I want to talk about how we can learn more about the disease, and will be the natural history study, which we call DECADE, Deciphering the CACNA1E related DEE. And the idea is really to get this information that probably you already know because you know your kids the best, is gathered together, and that we can first of all know more about the disease, but also know what we will be looking at once we need to do trials with the drug in patients. So the idea is really that you meet with your caregiving physician once a year for 15 to 30 minutes, and you fill in an online questionnaire, and we also will ask you nicely to also provide videos because it's quite hard to actually assess the movement of CACNA1E patients. So what we want to see is, first of all, does the disease work? How does it develop? Is there some patients that have a milder disease, which we already know that there are, which you also know? But we also want to kind of know how should we design the trials for the introduction of the drug. So how can you participate? there's some inclusion and exclusion criteria. So the patients need to have a CACNA1E mutation that is deemed pathogenic or likely pathogenic, and they need to have epilepsy, developmental delay, developmental regression, or movement disorder, and we probably need to exclude patients that have other causes that might cause this, which you can also contact us first to see if this is really an inclusion criteria. We will ask you for data on the genetic diagnosis, but also on the development in the first years and the perinatal time. And we also, what we really want to know is how does the epilepsy develop, how many seizures do patients have, and how does the motor development go on, and also how do your kids develop. That's really what we want to know, and that's what we want to ask.
Så et time-point of this, altså, og det er bare sådan, de forstørrelser af kinderne nu. Kan man det sådan noget?
Yeah, so people, patients who have died can also be included in the study. That's really important for us. So, of course, there's no yearly intervals then, but it's really important to us to know about what has happened and what was the cause of death. Yeah. So right now the study is designed, we have the questionnaires ready, we also really have the first approval by our ethics committee, but they had some comments, so we had to put it in another time, and right now we're waiting for the second ethical approval, and we hope that they won't have any more comments then, and this will probably be in June, July.
Should be earlier.
Yeah. So probably in the next. And then we're really happy to start the study, and we'll ask you to join. Yeah.
Okay.
Yeah, we're also waiting for this. Okay. With this, I think we're done. And in Tübingen, we thank all our collaborators and all the people in the lab that work on this.
So we can maybe say that Thomas Ott made the mouse.
Yes, yeah. Thomas Ott made the mouse.
With Michael Alba, we have also a new pediatrician, which is closely collaborating with us, which is important. Yeah, you probably know him. Or
yeah, we are treated by
exactly.
Dr. Alba.
Yeah, exactly. And then people from the lab are Jacqueline Barr, Patricia C, who left again, but now Melanie. I forgot Melanie. Madeleine, sorry.
Madeleine.
She was on one of the slides. She does the cellular recordings at the moment. Peter Müller did the study with the animals and Uli as well, and also will help with the clinic. And then there are of course the international collaborators that helped us to identify the phenotype. Yeah, so it was not our finding. We just did the functional studies. Of course, they are also important to get all the patients then together for trials and so on. So maybe Henning, you may